All right, in this video, we're going to look at myocardial action potentials. Previously, we have covered action potentials and pacemaker potentials of autorhythmic cells. Remember, we looked at something like this, where we saw the pacemaker potential leading up to an action potential once you hit threshold. And again, these are in autorhythmic cells, namely the SA node specifically, okay? What we're doing in this one is looking at a different kind of electrical activity, and it's going to look something more like this, where you've got that distinctive plateau. And this will belong to the cardiac muscles themselves, the cardiac muscle fibers themselves. So here are our learning objectives. We want to look at the different channels and that are opening and closing, and whether these are depolarizing or repolarizing events. We want to talk about that plateau in a little bit more detail, as well as why we need that plateau. And again, channels that are responsible for repolarization, and just differentiating between the things that we've already learned with what we're talking about right now, which are the cardiac action potentials. Okay, so this is your cardiac muscle fiber, and I don't really need to spend a lot of time on this other than to point out that you got a ton of mitochondria, as we might expect. But the main thing I want you to notice is these things should look familiar. These I bands and A bands and Z discs, sarcoplasmic reticulum, T tubule. You guys saw all of these things before when you learned skeletal muscles, and you talked about the excitation contraction coupling and the sliding filament model. Well, we just want to leave it at that and say it's the same stuff that goes on here. You still have actin and myosin. You still have the myosin binding onto that binding site on actin, promoting the sliding filament. It's still a muscle. So those things are pretty much the same. The difference, though, the meaningfully different thing, though, is in the electrical activity. So let's look at that. So what we have here is the action potential of a cardiac muscle fiber sitting at rest at negative 90 millivolts. And if we were to just read the graph out, if we simply read it, just put words to it, and then we'll get into some of the details, we say that at rest, something happens, and I now have a massive depolarization until I get to right about positive 30 or so. From there, I have a plateau where I sustain depolarization. So once I'm up here, you are going to sustain or maintain depolarization for a window of time before you quickly descend back down to resting memory potential in the repolarizing event. So let's take a look at that. When we look at our picture here, this is the cytosol, again, which is inside the cell. This is the interstitial fluid. And so what we're going to see from rest at negative 90, we're going to see opening of voltage-gated sodium channels. So which way does sodium go? When you open up sodium doors, this is something that you guys committed to memory. Which way does sodium tend to move based on its electrochemical gradient? Sodium always and forever will move from outside the cell to inside the cell when it's moving down its electrochemical gradient. And that's what we see here with this black arrow. Sodium is going from outside to the inside. And when that happens, we depolarize. And again, you can imagine sitting here at negative 90, and we take a positively charged cation, send them in, which would bring this to, let's say, negative 89. Another positively charged cation comes in, and now it's at negative 88, and so on and so forth, all the way to plus 30. Now, remember what I did say to you guys already in lecture. I said that it's not as if it's a one-to-one change. It's not as if one ion enters and leads to a 1 millivolt change. I'm only using that as an example just so you get this idea of dumping in positive charges, which is going to make the inside of the cell less and less negative, and then eventually increasingly positive, okay? So sodium comes in, and we get that rapid depolarization from negative 90 to plus 30. Is that what we see here? Yep. Sodium comes in, more sodium comes in, more sodium comes in, more sodium comes in, and eventually I'm all the way up to plus 30. Now remember what happens with these doors. When these doors are open, remember I said that they basically kick open for a brief window of time and then they immediately close. And so by the time we get up to plus 30, now the voltage-gated sodium doors are going to close, and these channels then close, okay? So the next thing that we're going to see is we're going to see that once you are at that peak of depolarization, we will see that your potassium channels will open up. But hang on, before that though, or really at the same time and more significantly, we see opening of voltage-gated calcium channels. Now, when you see opening of voltage-gated calcium channels, which way does calcium move? It'll move from outside the cell to the inside of the cell. So look at what's happening here. Potassium doors will open, and you see that potassium will start to leak out, but calcium channels open. So what you're seeing is you're starting to repolarize, but because calcium channels then open, then I sustain depolarization to here. Eventually, calcium channels will close, potassium channels stay open, and then potassium continues to move out, leading to repolarization. So let's do that again. You can read this here below as I talk it through, but I'm going to let my cursor follow through on this chart. So from negative 90 millivolts, voltage-gated sodium doors open, and therefore sodium comes from outside the cell to inside the cell, leading to depolarization. Once you get to the peak, sodium doors close, and potassium doors also open. Voltage-gated potassium doors open. I repolarize a little bit, but because I see opening of voltage-gated calcium channel, calcium comes from outside to inside, and we're seeing that right over here, calcium coming in, even though potassium's coming out. And because calcium's coming in, I sustain that depolarization. I maintain that depolarized state. Eventually, eventually calcium doors close. Potassium doors are still open. And remember that when potassium doors are open, potassium goes from in to out. Remember that? And we see that potassium is going from in to out. Potassium continues to go from in to out, leading to repolarization of the cardiac muscle fiber back down to resting memory potential. Okay, so this is another slide. It's showing you the exact same thing, opening of voltage-gated sodium doors, and then here a little bit of potassium opens, but we see calcium channels open, leading to sustained or maintained depolarization. This is your so-called plateau. From there, calcium channels will close, potassium channels are open still, and we see repolarization. Okay, so that's what we're seeing in terms of electrical activity of the myocardium. So now let's talk about why this is meaningful. When you look at this, this is a nice slide. This is showing you the the electrical activity. It's exactly the same thing that we just talked about. Here's the electrical action potential right over here, and this purple line is showing tension being developed within the cardiac muscle itself. And so as that tension is being developed, we see that eventually the tension then declines, right? So there's a contraction, which then there's a relaxation that occurs. And so what we see here, because of the plateau, what is the meaning of the plateau? Why do we need that? What that does is that it actually prolongs the refractory period. It prolongs your refractory period. If you can only say one thing about that plateau, you say the purpose of the plateau is to prolong the refractory period. Which kind of leads to the next obvious question, which is what is a refractory period? Well, what a refractory period is, it's that window of time. It's a period of time, okay, indicated right over here. It's a period of time where no matter what I do to that cell, I cannot fire off another action potential. One more time, a refractory period here is that window of time, as you see on the x-axis here, that no matter what I do to that cell, I cannot fire another action potential. That's a refractory period. Well, why is that meaningful? What that means then is the action potential is here, and remember that the electrical event precedes the contractile event in purple. Okay, the blue electrical event precedes the contractile event in purple. What that means then is that you cannot fire another action potential right over, let me draw a line here so you can see it. You cannot fire another action potential right over there. It just can't be done. You can't fire another action potential right over here. You got to wait till the end of that refractory period when I get back down to resting memory potential. So what we're seeing through this prolonged refractory period is an inability to fire another action potential until I get to here. Well, who cares? Why does that matter? It matters because when you have a prolonged refractory period, then you cannot allow contractile events to summate. It cannot occur. What does that mean? I want you to think about this, and this is where you'd have to go back and think about skeletal muscle and tetany. I don't have a slide on it here, but what is tetany of a skeletal muscle? It's sustained contraction. Do you ever want sustained contraction of your biceps? Of course you do. If you're holding your nephew in your arms, right, you're cradling your nephew with a bent elbow, then you want sustained contraction of your biceps. You don't want that thing to contract, relax, contract, relax, contract. You want a sustained contraction. If you're holding a tray of food or holding something, then you want to have the ability to sustain contraction. That is physiological tetany. In the heart, we do not want to even let that be an option. We cannot have physiological tetany in the heart. In other words, once the ventricle contracts, you got to let that thing relax. You got to give it time to relax. So, and this is the ventricle. When it contracts, you must allow it to relax. You cannot build up tension and then sustain tension in that cardiac muscle fiber. So that will be the equivalent of your heart going ba-boom, 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 and holding that contraction. It just isn't going to work. And maybe right now there's some intuition for that, but you don't quite see it. What I will tell you is that if you keep squeezing your heart muscle, then now you're not giving it a chance to refill and then squeeze again, and refill with blood and squeeze again, and refill and squeeze again. So ventricular filling is just as important as ventricular contraction to push out blood. And the reason is that, I mean, that's the whole reason we contract, right? We got to move blood forward by generating that pressure gradient. And so the prolonged refractory period prevents cardiac muscle tetany, which is sustained contraction. We don't want to allow that to occur. Okay. So when we look at this, we're looking at, again, pacemaker potential and what goes on at the algorithmic cells. We're looking at atrial muscle, and there is a plateau, maybe not quite as dramatic as ventricular muscle, but there is. Here's some activity of the AV node as well. Ventricular muscle, you see that long plateau, again, because you do not want to permit tetany of that cardiac muscle. And by having a long plateau, you prolong the refractory period. Okay, so describe the generation of myocardial action potential. Hopefully you are clear on that. Sodium comes in, a little bit of potassium goes out, and then calcium goes in, and then potassium goes out. All right. Which channels open at rest? We talked about that. Sodium will open when you're at rest, and that leads to depolarization. At the plateau, calcium channels open, and calcium comes from out to in. We need the plateau because it prolongs the refractory period, preventing tetany. Which channel is responsible for repolarization? Those would be voltage-gated potassium channels. Differentiate between pacemaker potentials, which we looked at right over here, and myocardial action potentials, which we looked at right over here. Or you can look at the difference on the same plot. There are your pacemaker potentials. This is the ventricular myocardial action potential. You can see they look very different. Okay, that'll wrap it up for this video.